Hello, this is Thomas Hartung from Johns Hopkins here in Baltimore. I'm the Doerenkamp-Zbinden Chair here at Hopkins for evidence-based toxicology. So you can imagine why I have a very, very deep feeling and interest into today's webinar. We are honoring one of the most important philanthropic donors to the field of alternatives to animal experiments, Hildegard Doerenkamp. And without any further ado, I would like to hand over to the president of the Doerenkamp-Zbinden Foundation, Franz Gruber, who is with us and will give you a bit of an idea about Hildegard Doerenkamp and her legacy.
Hello, thank you, Thomas. Goodbye, hello, everyone. I just want to introduce Hildegard Doerenkamp to you as a human, not a scientist. She was a layperson, but a very interested layperson. I will tell you why. Hildegard was born on the 5th of October 1920 in Cologne, Germany. She was a daughter of Wilhelm and Christine Doerenkamp, owners of a then upcoming company that produces recipe-free drugs until now. Mrs. Doerenkamp had a comfortable youth, was brought to school every day by a taxi or by the chauffeur of her father. After school, 1940, she wanted to study medicine, but her father didn't allow it. Instead, she got an education as a commercial artist and had to work in the company of her father. Ich kann es nicht vorwärts schalten. My PowerPoint doesn't work.
Dr. Gruber, do you have a set of arrows on the bottom of your screen?
Oh,
It goes. There you go. There you go. It's the delay.
Now, let us continue. During the war, the family stayed in Cologne, despite more and more air raids of the Allied forces. The young Hildegard Doerenkamp denied to flee in the air raid shelters. Instead, she laid down in the grass of her parents' garden. The shock waves of the bomb severely damaged her hearing, and her eyes were damaged too. She had to wear sunglasses for the rest of her life. Conversation with her were always conducted with pencil and paper, and the work of the foundation was managed over the years by an almost daily exchange of faxes. Years of living in best and comfortable circumstances followed: marriage, birth of a daughter. Ms. Doerenkamp said to me that these years until the 80s in the last century were her champagne and caviar years. But more and more, she didn't like this kind of living. Hildegard Doerenkamp loved the outdoors. She enjoyed farming and managed farms in the Lower Rhine area, Germany, in Graubünden, Switzerland, and then Nova Scotia, Canada. Looking back, Mrs. Doerenkamp always called the 15 years she spent in Canada, where she also gained Canadian citizenship, the best years of her life. Her Canadian neighbors, Anne and John, were very sad to hear 2011 of her sudden death road. She was a great lady with a zest for life and a great determination. We are very sad to have lost a good old friend. Hildegard Doerenkamp was dog lover par excellence, especially the arguments of Professor Gerhard Zbinden, whom she met first in 1982, against the use of dogs in toxicology, convinced her so much that she launched a foundation together with Gerhard Zbinden in 1985 to replace animal experimentation. It was very difficult to find a picture of her without dogs, and if so, other beloved companion animals were to see, like here. You recognize her? Yes, The lady with the sunglasses on that carriage is Mrs. Doerenkamp. If no dogs, at least some horses must be in her life. I personally met Mrs. Doerenkamp the first time in 1998 in Zurich. She was a great lover of Altex, and during the trip through Europe, she visited me in my Zurich office. I didn't know then that she had in mind to invite me to be a member of the board of her foundation. But in 2004, I got the Doerenkamp-Zbinden Award for my life work, and in the same year, Mrs. Doerenkamp asked me to join the board of her foundation. This was her right according to Swiss legislation at these times. That very hard working years, and I had to meet Mrs. Doerenkamp by fax and phones, and then at least 4 to 6 hours per week. And I had to visit her in Vienna, where she lived since 2001, at least 4 times a year, to explain her manuscripts and Altex that didn't convince her. Hildegard Doerenkamp, as I said, was an enthusiastic reader of Altex from the first hour. She asked a lot. Often a highly specific question even led one to question whether one had really understood all details of the subject oneself. She made no secret of her special love of dogs, and with the sovereignty of age, she flouted philosophical objection to preferring one species over another. She enjoyed baffling her discussion partners with her statement that she cared least about primates, they being too similar to humans, with whom she really did not get along at all. Though, of course, they never missed qualifying such statements with irony. She was also confident enough to laugh about herself. This area of the foundation can be called the area of the alternative chairs that we founded in this time after Hildegard Doerenkamp sponsored a private chair to the president of the Doerenkamp-Zbinden Foundation, Kai Brune, where she became honorary senator of the university. We founded the chair in Constance with Marcel Leist, Utrecht University with Bas Blauboer, Geneva University with Per Cosson, Johns Hopkins University, Baltimore with Thomas Hartung, and the Gandhi Gruber Doerenkamp Chair in India with Muhammad Abdul Qadir Akbar Shah. Hildegard Doerenkamp always discussed with me what should happen after her death. She never wanted to go in the hospital. She even didn't like the Seniorencentrum in which she lived in Vienna. She wanted to flee all these old people. So I moved with her during several years from town to town to Schweinfurt, Frankfurt, Constance, Friedrichshafen, Utrecht. She was unrestless, but full aware to the needs of the foundation and the chairs. Her last station before her death was on herself in Willingen in the Black Forest, where she lived by herself. Hildegard Doerenkamp prepared her passing in detail. They categorically refused intensive medical care to prolong her life. Mrs. Doerenkamp demanded to be buried at a small forest clearing at Kingsburg Beach in Nova Scotia, belonging to a farm. Here she buried her dogs, and here she wanted to be buried herself. And so we did. Our trip to perform this last mandate was a very melancholy one. Mrs. Doerenkamp's urn was buried at Kingsburg Beach on the same site where her dogs have been buried years ago. May she smile down upon the further progress of her life's work, the Doerenkamp-Zbinden Foundation. But, dear all, today is not her funeral. Today we celebrate Hildegard Doerenkamp's centenary. Hildegard, thank you so much for all what you have done for animals. Cheers.
Good. Thank you very much, Franz. I think this is was a very nice introduction and overview about her life and major contributions. And we would like to move on and showcast now the 5 chairs which are remaining, which are active and which have been supported by Hildegard Doerenkamp, the Doerenkamp-Zbinden Foundation. And actually, historically, the first was Marcelized with the chair in Constance. So Marcel, it's my pleasure to hand over to you and give us an idea what you have made from this appointment.
Okay, welcome everybody. Thanks, Franz, for the very nice setting the stage. Thanks, Thomas, for the introduction. Just brief question to Camilla. Is this all clear? Is my screen visible?
Yes, it's perfect.
Okay, thank you. So I even managed to incorporate some 100 years birthday, and this is what we want to show. I realized that also now, even around here, some people don't know the sign of the Doerenkamp Foundation. So I think it's also good to remind people of this. And 100 years is a long time. So I just wanted to show this. This is an image of Constance taken from the Zeppelin. And this is how it looks 100 years later. It's the same image of the Niederburg. We see that some things changed, and I hope that this could be symbolic for what Hildegard Doerenkamp also changed in the science scene. And for that, I am going to use 2 slides just to show a little bit what the ideas were of her and also the foundation at that time. So the classical way to support alternative methods was project sponsoring. And that was leading to the alternative method development. So then from the good projects, some method was indeed being developed. And then the next step was also to implement this method, because that point was to have the methods in use. And then, of course, they met with some regulatory hurdles, with also academic mistrust to alternatives. This was a bit of a strange field. Therefore, there was a lack of an audience and also a lack of continuity once the project sponsoring was left over. And this left the end result to be an academic publication. So this is not meant to be trash, but this is the recycling sign. So basically the recycling went back this side of the circle, not really to the implementation. This was the good outcome, and the bad outcome was the loss in translation. And we know also from a lot of these overviews that more than 90% of all the methods that were ever developed are just lost in this translation. So it was really a revolutionary move to rethink this situation and to think what could be done different. And this idea was sponsoring a chair for alternative methods. So what can be reached by this? So first, there is a sustainable project activity. It's not 2, 3, 5, or whatever year windows, but it's sustainable. And one has not only the chair, but the chair educates further people. So loads of master students, PhD students, postdocs, and also other people that qualify for new professorships come out of this. So there's an amplification of knowledge and specialists. And having a chair for this strange new field also was giving more peer recognition amongst the other professors. And of course, this was meant to enhance the credibility and the confidence in the field. And then professorship is usually part of a network of academic institutions. So this creates amplification and synergy, for instance, in European consortia, where many other professors, but also companies and regulators are in. And this can eventually then lead to the industry interest, because they might be part of such a network, and the application, also regulator interest and acceptance. And what is important, all these things can really not lead to very much if they stay national, but there need to be large networks for an international harmonization. So I picked this all from an article I wrote in 2006, what could be the use of a chair. And of course, it's up to others to judge whether these things have been reached or whether other things were perhaps more useful or some of them were not useful. But the idea as such was revolutionary, and for a foundation to go and to contribute to a university with a chair was a new thing. So we'll see how much this is being picked up in the symposium. I think I use my remaining minutes to give you a little bit of an overview how the work can look like in the chair and how we are interested in solving issues with teratogenicity. So we work on cell culture-based models, and the early brain development looks a little bit like shown here, and the neural tube has to form. If this stage doesn't work, so the closure doesn't work, and we know drugs and other compounds that prevent this closure, then some damage can follow. And we would like to use cell culture models to predict such damage. So we started working with pluripotent stem cells here very early, and they have certain markers. They can be differentiated to neural stem cells, so they have entirely different markers. And then these cells start self-organizing in so-called rosettes. So one can see many of these rosettes. And this self-organization is a little bit like the neural tube. And we can indeed see effects of chemicals that can disturb this process. This can be measured morphologically or by gene expression. And one can see what the challenges are. So this is this epileptic drug that actually causes these defects. And we can see, we can get quantifications. There's a clinically relevant range. There's a cytotoxic range. And of course, the challenge is a robust quantification method, like developing an index or some kind of scaling. The next challenge is to come up with a toxicological prediction model, which gives also performance parameters. It gives a no effect level. It has some range where things are happening. And finally, of course, these in vitro data need to be extrapolated to in vivo with all the toxicokinetic properties. And I'm just addressing 3 or 4 of the challenges this approach has at the moment. And one is to move from 2D cultures, as seen here in the neuronal cultures, to something that is 3D. And that would mean it's more like an organ. It involves different cell types, as we see here in green and red, glial cells and neurons, or it might even have a blood supply. Another challenge is to move from structural defects to functional defects. In neurodevelopmental disturbances, for instance, resulting in autistic spectrum disorder, the function is important. And that means also moving not only to morphological functions, but to physiological functions. So a morphological function would be that neurons have the capacity to grow neurites. And we have, for instance, set up systems for peripheral and central neurons. And that these neurites not only extend, but as we see here in the bottom, we can image the mitochondria moving within the neurites and take this as a functional readout. But we might also have cells that need to move to the right position. So if one scratches a cell layer, then the cells can move back, or they can be prevented in moving back. So these would all be morphological functions, but they would all not detect a compound like nicotine, which is known as a developmental toxicant. So for this, one has to be able to measure the electrical activity of such substances in the cultures, or the burst firing of a neuron provoked by such a compound, and then apply this to the screening of compounds. Here we see a quantification of the effects of nicotine and of other compounds that affect the same receptor. And here we see tubercularin. This is a poison that would block these effects of nicotine and that can be quantified. So this was only meant to give an example of what functional assays should be able to provide in information. And then another challenge is to move from a single assay, as we have seen, can be very specialized to a battery. And this was, for instance, formulated here, that there's an international effort to form a battery for developmental neurotoxicity testing. And an example is given here where we work with Ellen Fritsche from the IOF and EFSA, the Danish EPA and the OECD, to cover all kinds of different functions in the development of the nervous system, proliferation, migration, neurite morphology. And these are all covered by different assays in the battery. Then others contribute assays. So the US EPA is contributing some assays and the JRC is contributing some assays. And we hope that then such a complete battery could give a complete information. The outcome would look like this. So we tested 120 chemicals, and these are all the different assays, and the colors code where a compound does something harmful or not. And finally, the challenge is to move, of course, from academia to industry and regulators. And here's an example setting the stage for next generation risk assessment. So not just defining hazard, but going a step further in a large European consortium. And this is one of the outputs. So it's a method documentation, data processing, and testing pipeline for regulatory use. So this is an important transition. And with this, of course, CART comes into the game, and that forms the bridge from Constance to Johns Hopkins. And I think Thomas will pick up on this. And actually, we are forming a powerful triangle between the Doerenkamp Foundation, CART, and also Altex, which is an important publication medium. And that also shows how these test systems are being transformed into industry use and the benefit for drug development also recognized by regulators. So I hope I could give a short impression on what we are doing and also on what the initial ideas were in setting up the chair. And I think the timing should also be fine to allow the shifting. So thanks to the audience.
Thank you very much, Marcel, for getting the first chair represented so nicely and making some links to the legacy of Hildegard Doerenkamp and how she and the foundation developed these concepts of having chairs for alternative methods. And it is my pleasure to take over now from here and tell you something about what Baltimore is doing and how we are linked to the legacy of Hildegard Doerenkamp. So the, oops, I hope this is the right one. Yes, looks good. No, it's not. Sorry, I have to, it's the wrong presentation. Too many open. So here we go. So in here you see very faintly the Bloomberg School of Public Health, which hosts the Center for Alternatives to Animal Testing, CAT, for now more than 39 years. So we're looking forward to celebrating 40 years of CAT next year. And since 2009, we have the pleasure of having the Doerenkamp-Zbinden chair here for evidence-based toxicology. You already heard about Hildegard Doerenkamp, and what I think is quite important to understand is that she formed this alliance with Gerhard Zbinden, a very renowned toxicologist from Switzerland, who very early on started to criticize the use of dogs in toxicology. And by creating the Doerenkamp-Zbinden Foundation, a certain spin towards toxicology and alternative methods was implied with Gerhard Zbinden's legacy, who passed away in 1993. Gerhard is certainly one of the iconic figures in creating the entire movement for alternatives to animals. And I always like to cite in this context Isaac Newton, who said, we are standing on the shoulders of giants, and he's certainly one of them. But it takes a village to really get something done. And Hildegard Doerenkamp founded several of these villages. And from my point of view, the chairs she enabled are villages of working towards alternative methods. It is not just individuals represented here or heading as a type of mayor these villages, but it's the people behind it. My first personal connection with the Doerenkamp-Zbinden Foundation was in 1996, when I was awarded the Doerenkamp-Zbinden Award for a pyrogen test I had developed together with my mentor, Albrecht Wendel. You see us to the upper left. What was really remarkable for me at the time was that in the audience was Bill Russell, so one of the founders of the 3 Hours, and it was one of the 2 encounters I had with Bill Russell when we were receiving the Doerenkamp-Zbinden Award. This put us on a track, and it put us on a track both for the pyrogen test, and it put us on a track for working with the Doerenkamp-Zbinden Foundation and alternative methods in a broader sense. So it was really one of the changing points in both of our careers, I have to say, for Albrecht Wendel and myself. The pyrogen test was a driving force for really overcoming animal testing in this area. If you're interested about the latest statistics, they were published in February by the European Commission, and there's an article in Altex available. But what was really remarkable was that the use of rabbits for the pyrogen test, first of all, is enormous. You have to compare the 160, 170,000 rabbits which were used per year in Europe for these assays to about 5,000 animals which were used for eye irritation and skin irritation before any type of replacement took place. And you can see here now how the latest report shows how there is a continuing decrease from 2015 to 2017. So after some 25 years of work, we really achieved something. But I personally don't understand why we are not yet at 0. But it was definitively a very important component that this award, a recognition only half a year after this essay was published, and just around the time it became commercially available, was such an encouragement to move on here. Here you see Albrecht Wendel and a younger version of myself. And we joined instantly after also formerly the Doerenkamp-Zbinden Foundation. Albrecht served there until 2003, when I joined the board of the Doerenkamp-Zbinden Foundation, and we've been working since then. And the Steinbeis Transfer Center for In Vitro Pharmacology and Toxicology was our activity until 2006 by Albrecht. I had to leave it when I joined ECVAM, but it was one of the carriers of methodologies like the pyrogen test into many different laboratories. And it was then when, very much with a lead by Albrecht Wendel, the Doerenkamp-Zbinden Foundation developed the concept of a chair for alternative methods. And Constance was successful in becoming the first place, and with Marcel, as you heard, the first chair for alternative methods worldwide was installed here. There's an entire booklet which was produced in 2002 by the Doerenkamp-Zbinden Foundation, where they argued with several contributions on why impacting on education by creating academic centers is an opportunity here. Marcel and me worked closely together over these many years, and I have chosen these 2 articles which were in the same issue of Altex in 2008, because Hildegard Doerenkamp was especially excited about these articles. And you cannot imagine, she's a layperson, or was a layperson but she was really carefully annotating and reading all of our articles. She was really trying to discuss with us and trying to understand what this was. And these two articles for her were really a vision for how to overcome animal testing in toxicology. And she also gave us a very nice personal present as a consequence of enjoying these articles, which are really visionary. This is one of the figures of the Food for Thought article of the two. And it shows something which was, I think, for the first time really demonstrated like this. We said at the time, we should start looking at structures and see whether structures are unlikely to carry any activity. And I have to say, our work in 2018, developing an artificial intelligence tool to do exactly this based on automated read across, is implementing the vision of making structure a first step in assessing. We then came up with concepts, and actually it is, to the best of my knowledge, the first mentioning of internal TTCs, thresholds of toxicological concern, a concept which we took up in 2018 and which we will now be able to follow up in more detail in a just granted application by the European Union, the OnTox project, which will make tremendous use of thresholds of toxicological concern internally and as external dose. We have continuing both sides of the Atlantic to work on classical alternatives in vitro and in silico. So this is the, let's say, the business which took place. But then we were able also to develop the human toxome project, which was following the idea of if there is no pathway triggered, then there is probably no hazard, by mapping pathways of toxicity. And only most recently, together with Marcel, with his lead in Constance, we have applied also to create a program on biomarkers and pathways of and the kinetics, especially the kinetics, the PVK part, to address. And we hope by applying these different steps in a strategy to minimize the number of things we cannot call where the little red question mark remains. So I think this was a pretty good vision. And from this, the idea developed, we really have an opportunity to develop concepts outside of the pressure of regulating or being regulated as academics. So we set up a variety of programs, the Transatlantic Think Tank for Toxicology, sponsored by the Doerenkamp-Zbinden Foundation and others. And we also gave our vision into the public open. In this context, when I was offered a professorship at Hopkins, I was not really completely intrigued. But then Hildegard Doerenkamp said, Thomas, what do you think about endowing this as a chair? And this made the whole deal much more attractive. You see Alan Goldberg here, who at the time was about to hand over to a successor when Hopkins approached me, and I accepted the Doerenkamp-Zbinden endowed chair for evidence-based toxicology at the time. Evidence-based toxicology was an idea we had been pursuing since 2002, mainly with my PhD student, later collaborator, postdoc, and also in ECVAM, Sebastian Hoffmann, who wrote his thesis in 2004 about evidence-based in vitro toxicology. And so we developed the vision that evidence-based approaches, systematic reviews, meta-analysis, quality scoring, and similar stuff could actually help toxicology. And we organized the first conference on evidence-based toxicology in 2007. And then the chair in 2009 here at Hopkins gave us the opportunity to really install a type of headquarter to pursue this activity. Since 2011, we are formally hosting the evidence-based toxicology collaboration, which is a mainly transatlantic organization, but now increasingly also expanding to other parts of the world, trying to promote this concept of evidence-based approaches in toxicology. Marcel already mentioned that we formed also formally an alliance by creating CAT Europe, so that the two chairs here are really very closely linked. This is the former dean, Mike Clark, and and Dr. Rüdiger, at the time the president of the University of Constance, when this was formally installed at the University of Constance. A very close link between the two chairs. We cannot talk about work and Hildegard Doerenkamp without mentioning dogs, because she was always intrigued mainly by dogs. I find it always very amusing that at one point she said she's not interested in replacing work on monkeys because they are so similar to humans. This might be ironic, I hope so, but she was definitely very much into everything which could be done to replace dogs. And you can see both the US and Europe, they are clearly very much into pets, dogs as pets, but they also are using a little bit to a different extent dogs in research. This was the situation around the time when we, in order to honor Hildegard Doerenkamp, we did carry out 2 workshops on dog use in biomedical research. So this was the first time shortly after her passing that we were really trying to honor her by having workshops on systematically replacing dog use. They were planned before her death, but we only could carry out them after this. What is quite interesting is that there's a very different attitude towards dogs in the US and Europe. Here in the US, we have only about 60% of the population of Europe and only about 92% of the number of researchers. But we are using 3 times more dogs than there is in Europe. So dog is a much more frequent commodity in research in the US. Our 2 workshops are published as part of the T4 workshop series. We were very excited that this workshop in Budapest was actually covered in Nature. I think this is largely unheard of, that a scientific workshop leads to a Nature article, because one of the participants was a Nature journalist. This continued into activities, mainly with Dow Agrochemicals at the time, trying to help, because this was one of the key recommendations, to finally get the 1-year dog study for pesticide use, which was already abandoned by the EU and the US out of the legislation in other countries. Because interestingly, though Europe and the US said we don't need this test anymore, Brazil, Japan, and Korea did. We were successful very quickly to convince Brazil to abandon this. And we then managed also to get Korea to abandon this. So at the moment, there's still, however, unfortunately, Japan requiring this test, and we are continuing to work on this. So we are planning for next year the first virtual workshop on dog use for pesticides, because we want to make this really a test of history. So I would like to end here with my short presentation. The 100th birthday of Hildegard Doerenkamp, I think, is a very nice opportunity to show the power of scientific philanthropy. It shows that her legacy lives on. Her spin and focus on dog use and toxicology are something we are trying to put into practice and implement. And I think we finally see some light on the end of the tunnel for a real change, a substantive change in this part of science. Thank you very much. So, and then it is my pleasure to move on. I think according to the program, this should be Bas Blauber. Is this correct, Camilla?
Yeah, that is correct.
Good. My friend and colleague Bas Blauber from Utrecht, known to many of you as the editor of Toxicology in Vitro. He was actually the 3rd Doerenkamp-Zbinden professor in after Marcel and myself, to be created by the Doerenkamp-Zbinden Foundation. Bas, the floor is yours. Thank you very much for joining us.
Thank you very much, Thomas. I'll try to share the screen now. All right. Well, thank you for the introduction.
Dr. Blauber, we still not see your screen.
It's on my screen.
You gotta click the green button that says share screen on a Zoom
I
call,
did. Yeah.
and choose the correct window, and then click share again.
Let's see where it is then. Share.
There he is. We see it. Yep. Perfect. Thank you.
Well, good morning, good afternoon, wherever you are at the moment in this world of COVIDium. It's very sad that we cannot meet personally, so we have to speak to you like this. Thank you very much, Thomas, for the introduction. I was involved in the Doerenkamp-Zbinden Foundation ever since the beginning of 2000, I think. I was invited to this chair in 2007, and it started all in 2008. And at Utrecht University, we were very happy that we would be involved in this. We had a legacy in working in vitro methods, and also especially in the field of combining in vitro toxicology methods with kinetic modeling. As you have seen from the talks of Marcel as well as from Thomas, the field of toxicology is quite interesting in this area, because you will have the possibility to work on this in a very close detail, and it will help you also in the field of the toxicological risk assessment, which is one of the things that we have to deal with regard to the amount of chemicals that are around us. So if I take you back to the history of toxicology and the toxicological risk assessment, it's all about quantification of some measure that you can have from animal models, and with the help of all kinds of safety factors, it's then meant to lead to safety standards for human exposure or environmental exposure, whatever you want to show. The point is that this type of work has always been flawed by the fact that there is a lot of uncertainty in this, especially because you are always looking at apical endpoints in what you see in these animal models, rather than a good understanding of what you are talking about. Also in the talk of Marcel, you have seen that we now have a lot of possibilities to do very detailed work in the field of in vitro. So my plea is from the beginning that we would say that we would have the possibility of having in vitro methodologies to pinpoint what we would like to know about the mechanisms of action, rather than these apical endpoints. Well, this has led to then a change from the present situation, in which we have these 4 steps of the risk assessment process: hazard identification, dose-response relationship, and then compare this with the exposure, and finally to come up with a risk characterization. And these are the steps that are all involved in this, to the possibility to use in vitro toxicology. Well, this is one of my favorite slides, I always say. You see here a mouse in a glass, and you see, would this be in vitro toxicology? Because the literal translation of in vitro is in glass. Of course, we're now talking about things that we can see in plastic rather than in glass. And the possibility to do this was very much boosted by a report that came out in 2007 in the United States, and it was building upon a lot of work that was done on both sides of the ocean in the field of in vitro toxicology that set the stage more or less for a new way of doing risk assessment and toxicity testing, and that made the possibility to pinpoint on understanding the biological processes that are involved, and also the way in which we can talk about normal biological function rather than adaptive processes or morbidity and mortality. And this is a scheme that came out of that report that is very much leading to what we are doing at the moment. As you heard also from the other speakers, we now have a good understanding of what can happen with cells in terms of a change after exposure to chemicals. We know a lot of the processes that are going on there, and we can come up with all kinds of methodologies that we can use for this risk assessment nowadays. We have the possibility to determine a specific cytotoxicity, which means the possibility to kill all kinds of cells, or even to go to much more specific toxicity, which is determined, for instance, if you have seen from the talk of Marcel in the field of the developmental toxicology. What we have to do is quantify this, and always that's one of the issues that we need to do, to quantify this in terms of coming up with what we would call a response surrogate. And here you see what comes out of this. We can produce a lot of changes versus the normal situation in relation to the concentration of a compound in our in vitro system. And what we then can see, you can see on the right-hand side, is that you have to pick out the most important one, the most relevant one of these curves, and then make sure that you are in the field of no effect, A, or probably the effect that might be counteracted in the adaptive processes, B, or the real adverse effects that you can see in the area C. Next step that we have to do is to translate this to what is happening in the in vivo situation. And that's what's called in vitro in vivo extrapolation. And if you try to quantify this, you come up with the nice abbreviation VIVIVE, which in French probably means to be alert or something like that. And this is what we want to do here. We have a lot of possibilities to measure all kinds of in vitro testing and come up with EC50s or whatever you want to have. We also have the possibility to study a number of the ADME or kinetic processes in vitro. And this will then lead to the possibility of understanding all these processes. A warning is in place here, because we need to understand exactly what's happening in these in vitro systems with regards to the exposure of the cells in toxicology, in a toxicology relevant way. You can have the possibility that you add your compound to your medium and that it sticks for 99.9% to the plastic well. So you will have a gross underestimation of the toxicity if you try to evaluate these data to the in vivo situation. So you need to know exactly what would be the kinetics also in the in vitro system. And we spend a lot of work on this in the time that the chair was active in Utrecht, especially with the help of my successor in that field, which is Nienke Kramer, who did a lot of work and is still doing a lot of work, building on the possibilities that we had when the chair was active in Utrecht. Next step is that we need to make the step from the in vitro to the in vivo situation. And there we have the possibility to build upon these so-called PBPK modelings. And this is a schematic representation of this, in which you can see that we can build up the organism in blocks that are representing the different organs, and we can make the connection between these things and see how a compound would move through the organism. And actually these type of things are resulting then in a number of algorithms that are solved simultaneously in your computer. And this will allow you to predict the concentration of a compound at any moment, at any place, at any exposure scenario in the in vivo situation. And this was developed in pharmacology, of course, and pharmacologists are using this just to, in the way I just explained to you, to see how the concentration in different organs will change after an exposure. What we do is the other way around, to use these models and compare the in vitro concentrations in a relevant, for instance, in the brain organ, and translate this back to a compound amount or dose that is causing this in the in vivo situation. And this then leads to a new paradigm in risk assessment that is based on the hazard identification step, but now not in vivo, but in vitro. A step, dose-response relationships, in which we are trying to build up a in vitro system that is predicting a point of departure, and that then compare this with the exposure assessment, and then do the qui vive with the help of the computer modeling, and come up with a risk assessment that needs to be just as good as the in vivo situation would give you. And since we can work with in vitro models from derived from humans, and if we can also make computer modeling for the kinetics of the compound or based on the human physiology, these things are probably much better than the animal model. And we have seen examples of this in the literature that are very promising and are really used already, for instance, in the field where animal experimentation is not allowed anymore, like in Europe with the cosmetics. So this then resulted in a sort of scheme in which we will be able to build upon the possibilities of all kinds of fields, like structure-activity relationships, chemical properties of compounds, kinetics, in vitro systems, that would finally lead to a risk assessment system that is not relying anymore on animal experimentation. And this is, well, in use already in different fields, but not always accepted already by risk assessors in the regulation. And that is what we have to work on. Step by step, we get there. And this is what I would like to conclude now. We need to take into account in vitro biokinetics, as I said. We need to take into account the kinetic parameters to correlate this to the in vivo situation. And of course, we need to come up with very well described in vitro systems that can give you the possibility to come up with the proper surrogates. And then finally, make use of all the knowledge that we have in the chemical, biological, and physiological area, like QSARs, cellular biology, systems biology, the omics, etc., etc. And this is necessary because we have to deal with a lot of toxicological data that we cannot make with animal studies. So we need to develop these kind of systems. So my conclusion is that we can have more science with less or no animal used in the future. So at the end of my talk, I would like to thank very much the Doerenkamp-Zbinden Foundation for the general support of our work. It is continuing in the good hands of Nienke Kramer and her colleagues at Utrecht University after my retirement, some 7 years ago now. And I have very good memories of meeting once with Mrs. Doerenkamp in Vienna at the beginning of my professorship there. And with that, I would like to thank all the audience, and especially Mrs. Doerenkamp, in thankful memory to her. Thank you very much indeed.
Thank you very much, Bas, for another facet of the legacy of Hildegard Doerenkamp and how she helped with your work. Thank you very much for this very nice overview. We stay in Europe. Pierre Cosson in Switzerland was supported by the Doerenkamp-Zbinden Foundation to get his professorship started. Pierre, the forum is yours, the stage is yours. Please explain what you have been doing with the support by Hildegard Doerenkamp.
Okay, I hope you can all hear me as I just move to start this presentation. So before I start, is this okay for you? Do you all see the first slide?
Yes,
okay.
It's perfect. Thank you.
So before I start this presentation, which talks about antibodies, let me first thank the foundation. The Doerenkamp-Zbinden Foundation has been instrumental in creating the Doerenkamp-Zbinden Chair for Alternative Methods in Geneva. And we have engaged in a number of projects for replacement of animal experimentation. I will talk today about antibodies. Antibodies are everywhere, and if you want to get an idea about it, look at the latest newspaper or the latest news you heard. It's all about COVID. Well, antibodies are necessary to detect fast detection tests for COVID. Antibodies are used for treatment of COVID. And what we all hope is that we will get vaccines against COVID that will actually mean that we will generate our own protective antibodies. Before I talk too much about antibodies, let me talk just about science and share a few words about the life science crisis. We are living through a big crisis in science, in life science today, and this crisis is best illustrated by publication in 2012, where a company tried to reproduce 53 high-impact original studies. So they had all the necessary means. They had a collaboration with the original researchers to try to reproduce their results, and they reproduced exactly 6 out of 53 of the studies, which means that 10% of the published data was correct, 90% was not reproducible. This is terrible for science, because science is based on reproducibility. What creates this problem of reproducibility? One of the main factors is the quality of reagents. And the worst reagent is antibodies. There has been, for example, here in 2015, a number of studies that showed that antibodies are extremely variable, very low quality, and a reagent which is the most poorly defined used in science. One of the most striking facts is that 50% of the best antibodies, those that are sold by companies, do not recognize their target. And all these papers have called for better antibodies for research. And what is striking, and this is what I will explain during this presentation, is that the main problem of antibodies today is that they are produced in animals. And that we would make a great service for science by replacing antibodies produced in animals by antibodies produced in vitro, antibodies that are called recombinant antibodies. So the first part, I will describe what antibodies are. Then I will talk about recombinant antibodies, how they are developed. And finally, I will show you what we're doing in Geneva to solve this issue. So antibodies are produced by animals or by humans. After all, we are animals. What happens when you inject an antigen to an animal, here a mouse, is that the animal chooses among a large number of possible antibodies, the few antibodies that recognize this antigen. antibody producing cells produce these antibodies, and then you collect the serum of this animal. And this is the typical antibody used in research today. It is called a polyclonal antibodies because it's actually a mixture of antibodies. All the antibodies produced by the organism that recognize different parts of the antigen, all these different antibodies are mixed in a serum, and this is what you use. It's a very low quality reagent. It's a reagent which is poorly reproducible. You will never get 2 times the same serum. So if 2 laboratories use serum from 2 different animals, they get 2 different results. And that's where the problem of reproducibility stems from. Just a word about the structure of antibodies. It's important to understand some of the aspects of this presentation. Antibodies are composed of typically of 2 chains, a light chain and a heavy chain. And these chains are dimerized so that there are 2 heavy chains and 2 light chains for each antibody molecule. It's a rather complicated molecule. There is a part of the antibody which is variable. It differs between each antibody, and this is what gives each antibody its specific properties to bind certain antigens and not others. Now, what are recombinant antibodies? How are they selected and how are they produced? So recombinant antibodies are based on a new technology that was developed over the last 20 years, which is called phage display. Phage display is a technology which is entirely an in vitro technology. So everything that I'm describing to you now happens in a test tube, an actually small test tube, usually a 1 ml test tube. So what do you find there? You find a phage, which is a virus that infects bacteria. And the peculiarity of this phage is that it has been manipulated so that at the surface of the phage, you find an antibody. And inside the phage, you find the DNA sequence that codes for this antibody. So if you want to select antibodies that recognize your favorite antigen, you create a huge library of phages, 1 billion different phages. Each one of them has one specific antibody at its surface. Then you select the phages that bind your antigen. This is entirely done in vitro, which means you have perfect control of the conditions. And then you sequence the DNA fragments in these phages, and you can produce the corresponding antibody. This principle of entirely selecting in vitro antibodies that recognize a specific antigen has been awarded the Nobel Prize in 2018. The technology is widely used for the development of recombinant antibodies by the industry to produce huge amounts of antibodies, but it's absolutely not used in academia. The reason why it's not, and these antibodies are very high quality antibodies because you know the sequence of each antibody, so you can decide that you will produce antibody 1 and you have a pure antibody 1 and you know exactly what it is because you know its sequence. Or you can decide to produce antibody number 2 that recognizes the same antigen but a different part. If 2 different labs use the same antibody, they use exactly the same antibody with the same sequence and they obtain exactly the same results. So this is a super high quality reagent and super well-defined reagent. So if this is the case, why is it that today still recombinant antibodies represent 0.01% of the antibodies used in fundamental research? Because it's difficult, because the technology is a little bit difficult to develop, and because essentially all the technology that's required for producing antibody has gone to industry and not to academia. So it's easy once you have a sequence that you have found, this is an antibody as it looks at the surface of the phage. It has these 2 variable domains, and you can then reformat it and produce it in this format which is called a mini-body. And this mini-body looks almost exactly like a real antibody. And when you use it for basic research, it's exactly identical to a real antibody. Yet those are very poorly used because we're missing information about how to use antibodies. We don't have enough antibodies developed yet, and we don't have facilities to produce these antibodies. So what we have done in Geneva, in the frame of the Doerenkamp-Zbinden chair, we have organized a facility which is open to all the academic world and which gives access to recombinant antibodies. The first thing that we have created is a database. This database contains a list of all the recombinant antibodies that exist, that have been characterized. We have added to this database all the antibodies that we have discovered, because our facility also does the discovery of antibodies, and it also sequences existing antibody producing cells, so that this database today contains 21,000 antibodies. We estimate that this is approximately half of the existing recombinant antibodies that are listed in this database. If this database is to be used by researchers, it's of no use to tell them that an antibody exists against their protein. We have to give them access to this antibody. So what we have created is a facility for production of these antibodies. Without getting in the details, what we're doing is that anybody can order any antibody from the database, and we will produce a small amount of this antibody and send it to the laboratory for a very small cost, just a production cost. And what you see here is the number of antibodies that have been produced over the last years by the facility. It's now nearing 1,000 antibodies. So for simplicity, let's say that this facility has replaced the use of 1,000 rabbits. And it has actually made the life of researchers easier because it has given them antibodies that are much higher quality than what they would have obtained by using animals. The second problem that people encounter when they find a new antibody is they don't know how to use it. So once you produce this antibody, it's very useful if people describe how this antibody really works in their hands. And for this, we have actually created a journal, a new scientific journal, fully open access. It was created 2 years ago now, and since then there has been 110 articles published. And each of these articles describes how one specific antibody can be used to do, for example, immunofluorescence or ELISA, or how can it be used in any of the possible techniques where antibodies are used. And this is then used and fed into the database so that antibodies now have their characterization attached. This is the circle that will make a difference in the field of antibodies. A database where every antibody is described, a production center that gives access to everybody to all these antibodies, and a journal that describes how these antibodies can be used and how they should not be used in some cases. I'm already getting to the end of this presentation, and I want to give you some perspective. I think that what we have created here, a database of more than 20,000 antibodies, a production center that has already produced more than 1,000 antibodies, and a journal dedicated to the description of these antibodies, is the essential mechanism to allow the academic world to shift from antibodies produced in animals to antibodies produced entirely in vitro. When I look at this, I think, well, this is not so bad, 1,000 antibodies, 1,000 rabbits, but frankly, I'm not satisfied. I mean, we know that the real numbers are hundreds of thousands. So we have now produced 1,000 antibodies, but we're only at the very beginning of this project. This whole project should now become bigger, and either with antibodies produced in Geneva or with a copy of this system in other places, we will have the capacity of replacing fully animals for the production of antibodies. And we know that this would reduce the numbers of animals used in experimentation by several hundreds of thousands every day, every year in the world. This is our dream. Thank you very much.
Thank you very much, Pierre. I think it is fascinating to see the recombinant antibodies and their various uses and how much you have been driving this story. This also resonates very nicely with the recommendation by ECVAM and the European Union to abandon animal-based antibodies, and especially the ascites mouse at the moment. I think this is really a very timely contribution. Thanks a lot.
Thank you, Thomas.
Last but not least, the fifth chair to be presented is in India. Mohammed Akbar Shah has been impacting very much on India and its use of animals, especially in education, but I don't want to take anything away. Thank you very much, Akbar Shah, for joining us here. It's always good to see you, my friend, and thanks for doing this at a very unusual time of the day. The floor is yours, please.
Yeah. Camilla, I am not getting the full screen.
If you scroll over the Zoom call, you should see a share button on the bottom. See share screen?
Yeah, share screen, yeah.
Okay, then click on that and then choose your presentation.
Okay, right.
And then you have to click share again because we're not seeing it yet. And it might be a little delay, so.
Let me wait for that to come.
Yeah, it's not coming. So if you click share screen, then you have to choose the window that it shows you, and then you have to click share again. It's like a blue button. Yeah, I still not seeing it, Dr. Akshab.
Yeah, it's just come. It's coming.
Yes.
Here it is. Yeah.
Yay!
Yeah.
Thank you.
Thank you for guiding me right into the PowerPoint. Yes.
No, that's great. Yeah. So you just need to go into from the beginning. Just press that.
Yeah. Yeah. This is the first slide, and I think I start speaking about my. Well, it has been a wonderful experience. Very nice time to be looking at all my friends, whom I have been associating with over the past 10 years, starting 2010 to 2020. So, Franz Gruber, Thomas, Marcel, Bas, Pierre Coulson, then some of the board members who are not actually seen, but they are still listening to and viewing the presentation. Camilla, it's been really nice to be sharing the screen on this day. Well, it's nostalgic to be remembering Doerenkamp. I have not really seen her, but had a very, very great lot about Doerenkamp from especially Franz Gruber, and to a certain extent from Thomas. So she has been a very wonderful person, and I used to be feeling very, very a person of pride to be working for Doerenkamp, particularly in my country of India. So I was invited in 2010 by France through one of my India contacts, and was asked to become the Doerenkamp chair in India. And a chair in India is something unusual, and, which is very popular in Europe, but not in India. So I had to spend some time to find out what really a chair means and what a chair would do. So I accepted the offer and went through half a century, half a decade, 5 years, a little more than that, with the foundation, doing a great lot of work, translating their vision into action. Well, we have the
Doctor, I'm sorry to interrupt you, Dr. Albert, but it's not moving. So do you want me to just share from my screen and then you can just say next?
you can't move it from your side, is it?
Okay, just give me a second. I'm going to share then. Because
you please try, if you can move it like.
Sure.
Yes. Is that right?
There you go. Can you see that?
I can see that.
Okay, wonderful. So when you need me to move to the next slide, just tell me and I'll move to the next slide.
You please now move to the next slide. Well, when I was asked to do this job as a chair in alternatives in India, I mean, as I said, I was not knowing much about what a chair should do and what I should be doing is in terms of alternatives. So I had some guidance coming from France and Shirani. And initially what we did was to start off 2, 3, 4 activities. And one was to establish a center for alternatives to animal experiments in India at Bharatdasan University. What you see now, the thing is one of the fascinating and fantastic campuses within Bharatdasan University that was brought up, established with the fund that came from Doerenkamp-Zbinden Foundation. So this is named as Mahatma Gandhi Doerenkamp Center, and the building is named after Doerenkamp as the Doerenkamp Enclave. So you have the Mahatma Gandhi, whose name is there in the takes part in the center, and Doerenkamp. And this is the building today with all green, lush vegetation going around. And of course, we will go into the building in the next slide. Next slide, please. And this is the center, the physical structure. You have the main building with the 2 ground floor and first floor. You have a lot of labs. I'm not going to the details. Very many facilities for in vitro research, in vitro activities and training, in silico activities and training, some alternative or model organisms like Caenorhabditis elegans, zebrafish, and hydra, and seminar hall and kind of all kinds of things that are needed to have it as a training come educational center. Next slide, please. And we had, I mean, because of shortage of time, I go piecemeal like how we went about doing things. My friends all earlier who spoke, they were asked to be concerned about very focused areas of research, whereas that was not the case with the India chair. In India chair, it was more a role to popularize, conceptualize the alternatives throughout the country, the huge country, and that was the big challenge. And we had our own plan as to how to go about it. So we started moving into programs, very important programs across the country. And one of them was the Indian Science Congress that was held in 2011, 99th Indian Science Congress. And it was very difficult to happen in a country like India to have a special session on alternatives in a Science Congress program. Has been really very difficult to have brought up. I had some friends and senior people like Dr. Ramamurthy and others who were able to arrange this for me. And we had speakers from Europe, like Thomas and all friends from CART, and they were there, and they had a good program. And I mean, here I should particularly say why I have a love for in Doerenkamp, Mrs. Doerenkamp has been that, unlike all other organizations which are concerned with alternatives or non-animal methods, who were all essentially concerned with animal rights organizations. Here, the Doerenkamp-Zbinden Foundation was practically an animal welfare organization, emphasizing the 3 R's in science, and with importance given to replacement alternative. So that has been essentially academic, unlike the other organizations, where it is more activity towards animal rights. Here it is animal welfare and propagating the idea, make people practice the concept and the methodology. So we did a good program in Bhubaneswar with friends coming from Europe, and it was a wonderful thing to happen. And again, we participated in the 2014 program, where again, we have done good job. And in the 2011 program, we had a special all-text proceedings covering our presentations, our talks at the Science Congress. Next, please. And so, having said that, the modalities we chose to emphasize in this country has been to target those people who have been using animals in enormous numbers. And the one which immediately draws the attention is the dissection and small experiments, physiology experiments in the undergraduate and postgraduate courses, programs in colleges. And you should believe me, easily about 50 million animals per annum are used in these dissections and experiments. And therefore that became the target of attraction for us. So we chose a methodology. We have a team. We had a team, and the team went around to places, the whole country, to colleges, university departments, had programs of sensitization, and then give them technical talk, then give on-screen demo, hands-on training, open house, so on and so forth. So you can see here 73 workshops conducted over a period of 6 years in the whole country, and it has been really a very nice experience. And so that was really a successful. That formed the groundwork to be speaking to the regulatory authorities and the students and teachers to change to non-animal methods. Next, please. And the outcome of this is cut short, but a lot of activity over 3, 4, 5 years. And ultimately, with the help of our supporters who are with the NGOs, like People for Animals, like PETA, like HSI, they were doing a lot of support to us, but we were the only academic persons associating with the whole movement. And we did a wonderful job, and we pressurized the University Grants Commission, the topmost regulatory authority of higher education in the country. So we could convince them to bring about a ban of use of animals in dissection. So that was finally ordered in 2014, and we have a GO that came out of it, and that essentially prevented anybody to use animals in dissections in any institution across the country. So it is roughly estimated to be about 30 million animals per annum. So that many number of animals is saved. Next, please. And if basic science institutions are using huge number of animals, similar is the situation with the pharmacy colleges and the medical colleges, which also use a huge number of animals in particularly laboratory exercises pertaining to pharmacy, pharmacology, and anatomy. And so we also went around the medical colleges and pharmacy colleges, where again, we had guests coming from US and Europe. We did a good job speaking to people, and it was really rewarding. Next slide, please. And as a consequence of this, like the dissection issue, University Grants Commission coming in with the regulatory announcements, the Medical Council of India and the Pharmacy Council of India, that was in 2014 in the respective Gazette notifications, came up with the announcement that there won't be any more animal use in physiology and pharmacology laboratory exercises, but rather they will do computer-assisted experiments or modules. Well, this is something which is something overwhelming, really. We were not expecting this decision, an announcement to this extent, but we were happy that Doerenkamp-Zbinden Foundation had a great role in these great regulatory changes. Next, please. And when that be done with regard to dissections and animal use in small experiments, and we knew that great many animals are used in toxicity testing and drug development in industry and in institutions in basic science also. So that was another target for us. So we emphasized on in vitro toxicology, in vitro methodology, in vitro approach to toxicity and drug testing. So we emphasized it, and we had our own lab. We conducted very periodical workshop, 21 workshops, and about 300 participants over a period of 3 years. And people had come from different states of the country to the place where the institute is located. Had been a very, very thrilling and very satisfying experience. Thomas visited us one of these workshops, and he was blessing the participants. Next. And that was not enough, like simply a 2D culture, flat culture, without any cell interaction was not so convincing. Of course, the beginners were happy, but people who are in more advanced level of research, they were asking for higher level of tools. So we brought in people like Albert Lai from USA, who is an expert on integrated discrete multiple organ co-culture. I mean, of course, today we have our 3D organ on chip technology, but before that, Lai's technology of 4 or 5 different kinds of cells put together in a culture medium, different wells, was a really attractive endeavor. So we brought him, and he gave training to people. We did some training in silico approaches. So we invited people from US again, and we have workshops done on that. Next. So the outcome of this endeavor, this portion, has been that the government of India notified that cosmetic testing should not hereafter be done using animals, a kind of situation which was announced in many countries in European Union. So we have a prohibition of testing of cosmetics on animals, and then prohibition of import of cosmetics tested on animals, which also came. So this is another huge jump towards saving animals from being sacrificed, used in experiments and sacrificed. So another milestone. Next, please. So, and if animals are not to be used in the experiments, in the cosmetic scenario, then what will they do? Well, OECD guideline is to be practiced and go for in vitro approaches through cell culture methods. And to sensitize the people towards those alternative methods that are available, we invited Dr. Christian from L'Oreal, and he came around and was giving training in many places. And we are so happy the technology has become very popular. Next. Next, please.
That was the end of our, I only have the 2 sets, so let me just start the other set.
Pardon?
That was the end of the first set. So let me just,
okay, you are going through the split one, then you have to open the second, right?
Yep, I'm
Please.
using the second, yes, just a second.
Yes, please, 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 please. And I think I am shooting to somebody else's time. So having done about as alternatives to dissection, alternatives to animal experiments, alternatives to animal use in drug testing and cosmetic testing, and also I had a little interest. Some research activity should go on. We had young people who were enrolled as fellows in the project, and they were helping us in the other activities. So I should help them with the fellowship and degree to take with them. So next, please. I'll be quick. So this is a girl, in the early days, what we did was, instead of going into advanced technologies, our effort was, our modality was to tell people that all that you can do using in vitro methods, in vivo methods, animal experiments, can be still done with non-animal methods and in vitro methods. So we collaborated with the chemic scientists whose compounds needed to be tested in animals. So we got them tested on in cell lines, and they were very happy with that. So we had a thesis, and we had very good papers coming out of it. The girl you see is Divya. Next. And here is Gayatri, another girl who were trying to spend time on using in vitro methods for finding the toxicity of fungi, with the fungal toxins. And again, beautiful papers and her thesis. Next. And this is Mohammed Zishan. He was a very interesting fellow. He wanted to do something new, not others have done so far. So he chose a very wonderful model, Hydra, a diploblastic animal, very simple animal. And he developed this into an alternative test model for chemical risk assessment and drug development. He made two wonderful papers with advanced technologies coming up. And this is his thesis, and this is apoptosis, which he was able to demonstrate in the Hydra. Next. And this boy is another interesting fellow, a young man who joined me, and he was also wanting to do research on Hydra. And he went developed further on what Zishan has been doing, and he has come out with more advanced applications of Hydra as a model organism in toxicity testing and risk assessment, and he submitted the thesis. Next. And this boy is Balaji. He had a different vision altogether, like not repeating the research or the methodology which the others were adapting. He wanted to bring up a newer culture method. So he brought up three-dimensional culture, we call it 3D spheroids, and he used what you call the substrate-free, a kind of using egg white as a substrate for culturing the cells. It's just a wonderful approach, and he also had very commendable publications and his thesis. Next, please. And to last is Karthikeyan. He was interested in silico method bioinformatics approach towards tackling the issues of toxicity and drug discovery. Here again, with beautiful papers, very commendable papers, and his thesis. So next slide, please. Putting together, we had 6 scholars who submitted each one a thesis, so 6 PhD theses. And we had 14 publications coming from these 6 theses. And beyond that, we had our collaborators who are bringing in chemicals and toxins for test in our lab. So we are a part of that research. So that is 87 publications. And we have another 4 publications in a different way. So totally 120. We have 20 reports came in Altex, and we have 9 review chapters. So there has been a wonderful thing as far as the research component is concerned, which was not really expected of me as chair to be doing in India. In India, it was essentially stop killing animals. That was the aim with Doerenkamp, and France were guiding me to, but still we did some research, and we are so happy with the outcome. Next, please. And we had like very wonderful moments during this happy journey. And one of them is our eighth Well Congress participation. We had a Best Poster Award, and that was really a wonderful experience. Next. And we had another award that came in the 9th Congress at the Prague, and there was an award from Japanese Society for Alternatives to Animal Experiments. Wonderful days. Next. And all these, I put this to last, all these achievements, 50,000 animals, 50 million animals per annum saved from all these exercises. Dissection band, in vitro approaches, all put together. My estimate is about 50 million animals saved per year, 6 PhD theses, 114 research publications, all in very good journals, all out of the money which came from the Doerenkamp-Zbinden Foundation, which Franz, Thomas, and others were very generous to provide to Mahatma Gandhi Doerenkamp Center, and that is 450,000 euros, and another 550,000 was cut together. So it is 500,000, which is equivalent to 4 crores of money in Indian currency, and the outcome, the productivity out of this is equal to several times of this money. And I should very specially thank my colleagues and my supporters, my students, and the foundation for all that they've done. Next, please. And next level was that before the project was coming to a close, we wanted all this research and activities to continue. So we moved with the Government of India through our appropriate contacts, and we have now the National Center for Alternatives to Animal Experiments. This center is now run in the Mahatma Gandhi Doerenkamp Center building, what you call as the Doerenkamp Enclave. Next. Next, please. And we have a society established, very good. And we have, these are my colleagues who have been voluntarily, without any honorarium, supporting me in all our activities in different ways, lectures, demonstrations, workshops, so on and so forth. Next. Next. And great lot of people supporting us in this activity. And I thank the France, I thank Thomas, I thank all other friends, my fellow chairs, and all my colleagues, all the colleagues who are helping me in running the center to what it is today. And I quote Albert Einstein that we should be compassionate to all creatures. Our task must be to free ourselves by widening our circle of compassion to embrace all living creatures and the whole nature and its beauty. This is not just for the sake of quoting somebody. I have 100% strong faith in Albert Einstein's this opinion. And so telling this, I thank once again all my friends from the Doerenkamp-Zbinden Foundation, especially Franz Gruber and Thomas Hartung, for their wonderful support in this endeavor. Thank you very much, and perhaps we will have more occasions to listen, see each other. Fine, thank you.
Thank you very much. It is really impressive how much can be achieved with a reasonable investment into this, and we very often look too closely, too narrowly into what's happening on the both sides of the Atlantic. I'm really appreciative of all the work you've been doing and adding this to our symposium. Thank you very much. So last but not least, we're going to talk about awards. And I hope I have a little surprise for some of you. Let me see here, share. Good. The Doerenkamp-Zbinden Foundation has, from its beginning, or at least very early on, started to award individuals and also groups by giving the Doerenkamp-Zbinden Award, which is an award which has been very diverse. Sometimes it has been honoring a lifetime achievement. In other cases, it has been really for a development, like in my case for the pyrogen test in 1996. As you can see, the last award was actually given out in 2011. The focus of the foundation had been on other things. The financial situation also dramatically changed after the passing of Hildegard Doerenkamp, so that this award had not been used as an instrument, but in recognizing today the achievement of the different chairs, and also seeing that this is an instrument for the future which could help us to engage young researchers as well as acknowledge people of a scientific stature which have made a difference in their career, we want to reactivate this award. So the first thing is, without a bursary in this case, we want to award the 5 chairs for alternative methods which have been created and have, with their work, which was documented so nicely in these 5 presentations, the 2020 Doerenkamp-Zbinden Foundation Award. So congratulations to my colleagues. We just wanted to have you on the list as major ambassadors for alternative methods for the foundation. However, we want to go a step further. And I think this is interesting news for many of you on the call. We want to reactivate the award, and we are aiming to award the Doerenkamp-Zbinden Award, which is typically bestowed with a 25,000 Swiss franc amount. All of this is at the moment under discussion, but we want to award this at the 11th World Congress for Alternatives to Animals and Use in the Life Sciences. As you all know, this has been postponed to August 2021 because of the pandemic. We are at the moment discussing the details. I'm personally very much in favor of a 2-step process. It is possible to nominate or self-nominate for this award. I would suggest that we invite letters of nomination of 1 page and would then come back to the contendants, those which the foundation board selects, to invite for a more detailed competitive description of the work to be funded. We are planning to get this out before the end of the year. In order to stay tuned, please subscribe to our Catwalk newsletter, which is possible through our website if you are not already a recipient, but also feel free to send an email either to Camilla or myself to get registered. Anybody can apply from each country in the world. Can be for in silico work, it can be for in vitro work, it can be for political activities. It is an award which is completely open to what is most convincing and what convinces the board of the foundation to actually put you into this honorful list of recipients. So I hope everybody liked today's event. I enjoyed it very much. A lot of reminders of what we have been seeing over the last 10, 15 years at the various places, the enormous achievements which a relatively small foundation could do. I think Hildegard Doerenkamp really lives on, and this shows you how philanthropy can make a difference. I would like to close with this. We have not foreseen a Q&A because this is a series of keynote lectures for to honor Hildegard Doerenkamp. I'm sure she would love this event. And I hope that from cloud 9, 17, whatever the number, she is looking to us and is saying, I did something which has a lasting mark. Thank you very much. And I hope to see you at another webinar CAT is sponsoring. Thank you very much.
Thank you, Thomas.
Well done, everyone.
Bye.
Thank you, Joe. Thank you.
Ciao, Franz.
Bye.
Bye, all my friends.
Akbar, ciao. Bas, ciao, Akbar. Tell.